Hey there, in this video I'm going to talk through how to lay out a dihybrid cross. I'm going to use the example of the peas, and in this type of pea we have the yellow and round pea and the green wrinkled pea. Yellow is dominant to green, and so we're going to give that a big Y there and little y for this green here. Now, since there are two alleles and we're going to work with the homozygous dominant peas for the yellow one and the homozygous recessive for the wrinkled, so we've got big Y and little y for the green condition. Round is dominant to wrinkled, and since this one's going to be homozygous, we will have two big R's inherited, and wrinkled will be little r, little r. The gametes produced then, so we've got big Y, big R, big Y, big R, and since those are the only options, we don't need to write lots of them out, so we'll just do big Y and big R like that. Likewise with the wrinkled pea, little y and little r, or little y and little r, so there they are. So this was the parental generation. And when these gametes cross, we have this seed being produced. So normally in a monohybrid cross we would do a Punnett Square. So we can kind of do that with this as well. We know that this gamete produces big Y and little r, and the other gamete is little y, little r, and it's as simple as that. And we combine big Y and little y, big R and little r, and that is what all of these gametes are going to be, or all these seeds are going to be. And that is the F1 generation. F1 generation. If we want to represent our heterozygous seed as chromosomes during meiosis, it might look something like this. So we've got a chromosome, or homologous pair, and we've got big Y, this big Y, and little y, there it is. And since we've got two chromosomes working here, because there's two alleles, dihybrid cross, with big R on a different chromosome, there, let's put them in place, and a little r there. However, because of Mendel's law of independent assortment, they could be the other way around. So we got a possibility of having that y on the little y on the other side, and there's a big Y, and let's leave that big R there, little r. So what's our possibilities then? During meiosis, when these get separated to either side of the cell to produce the gametes, we end up getting big Y and big R, these guys, and then these two coming together as little y, little r, and then this possibility would be little y and big R, and this possibility is big Y, little r. So 4 different gametes produced because of independent assortment. So let's do our cross. Again, it's a Punnett Square, but it's a larger Punnett Square to compensate for the extra gametes. And we'll have our male here. It's good to label sex of the cells, regardless if it's plants or animals. That way you can do sex linkage later on. So let's put our gametes in place. Let's start with big Y and big R there. We'll do big Y, little r, and little y, big R, little y, little r. And for the female as well. Alright, so what are we getting? We've got big Y and big Y in our cross, big R, so that's homozygous dominant for both conditions. Big Y, big R, little r, big Y, little y, big R, big Y, little y, big R, little r. All right, I'm going to pause and finish this off, and then we'll talk through it. All right, so I've done the majority of the dominant color characteristics for the yellows, and I've now got to one of the green seeds here. So little y is homozygous recessive for color, so we're going to get green, but we're still getting the homozygous round condition on this seed. So let's finish them off. We got little y, and big R, little r, so still dominant for the texture. Big Y, big R, little r. Big Y, little r, little r. Okay, so now, like here and here, we're getting the homozygous condition for the texture, which is wrinkled. Let's finish this one. So little y, green, big R, little r, so smooth and round, and little y, little r, little r. So finally, we're getting the homozygous characteristics for both the characteristics, no, homozygous genotype for both the characteristics. Therefore, phenotype, green and wrinkled. All right. So now let's count them up. So we've got the phenotype of round and smooth. What am I talking about? Yellow and round. And here we've got green and round, yellow and wrinkled, and green and wrinkled. Okay, let's count them up. So yellow and round, 1, 2, 3, 4, 5, 6, 7, 8, 9 of the 16, and there are 4 by 4, there's 16 of them. Green and round, 1, 2, 3, 3 out of 16. Yellow and wrinkled, 1, 2, 3. And green and wrinkled, 1 of 16. Now, this is typical phenotypic ratio for dihybrid cross of 9 to 3 to 3 to 1. Now, when Mendel did this, he got numbers that look something like 315, 108, 101, 32, and that comes out as roughly 9 to 3 to 3 to 1. And when you see this ratio, that means you are working with typical dihybrid inheritance. All right, time to give it a go.